Okej, så välkommen alla sammen. Den forelesningen skal være på engelsk for at jeg fortsetter litt sjenert på norsk og ikke så flink, men jeg forstår mye mer, så hvis du har spørsmål, bare stille dem i løpet av forelesningen. Also, this presentation will be recorded and then uploaded to YouTube, so make sure that you are in accordance with that when you are making questions or posing questions. Yes, so I should start the presentation now. All right, so about myself, so I will just speak a bit of myself, but before I would do that, there will be a game, and I will just require to grab a pen and pencil or and a paper, because you will require that. And so about why you are doing it, I'm just going to tell about myself a bit. I was graduated in Hungary in 2015 as a medical doctor, and I did my PhD, same in Hungary, in the topic of neuroscience. Then I spent a short period in Aachen, Germany, to study fMRI basics, and also spent a few months in Belgium, Leuven, to do some optogenetical manipulations in monkeys. And here I am in Motion right now, working as a postdoc in a project called ADHD, adult ADHD II, and how we are specifically aiming to check how the brain is working in adult attention deficit hyperactivity disorder. So I assume that you already got your or your pencils and papers, and I would just the instructions are to repeat the number of sequences you hear in the same order. So you're going to hear number sequences, and I would just ask you to write it down on the paper. So the first is 9, 2, 4, 5, 6. The second is 5, 7, 3, 2, 9, 1. The third one is 4, 8, 7, 5, 6, 2, 3. The next one is 1, 4, 3, 7, 8, 5, 9, 2. The next one is 1, 8, 3, 4, 5, 6, 8, 5, 6. And the last one is 7, 8, 6, 4, 3, 2, 5, 7, 9, 1. I'm pretty sure you all got that. And here are the correct answers. I would just invite you to go to menti.com and use this following code. You can check your answers here, and please just to vote what was the longest number of sequences that you achieved to repeat correctly. So I would just wait until you correct your answers. In the meantime, I would just open this menti.com and waiting for the answers. Oh, perfect. The first vote was achieved over the 10th. It was impressive. Okay. Well, this was a very tiny experiment, of course, but as you can see, we have quite a if we were to get more subjects, we would get somewhat similar. So I would just expect it to have around 7 or 8 correct the longest numbers. Yeah, this was the first game. It would be another one, so please don't throw away your phones, nor your pencils, nor your paper, because you're going to need it later. But let's just get back to the presentation. So the task that you were doing, it's called the digit span task, and it's commonly used neuropsychological test. And it refers to measure this short-term memory. And now short-term memory refers to a very short, as it says, it's a name, memory that lasts a very short period for 20 to 30 seconds. And it has a limited memory storage. Usually it was set up to that normal people would have 7-ish correct 7 memory storage. And as you can see here, the more memory items you would try to memorize, the lower accuracy you would achieve. I'm going to talk about this graph a bit later on. And for now, let's just go back to the brain and let's investigate how the brain works. So within our brain, there are several cell types called neurons. Of course, this is an enlargement of one neuron. One neuron is certainly much smaller. But these types of neurons, they produce small, short electrical pulses called the action potentials. And if many of these neurons are working simultaneously and synchronized way together, then what you would see is kind of this. So each point represents one small electric pulse. And so if these neurons were to align together, they would just produce a summation of these electric pulses that would represent in synchronous activity called brain oscillations. So these are the brain oscillations that we are going to talk about in the next few slides. And so the brain oscillations can be categorized by their frequency band. And one of the very most known is the Theta oscillations, which refers to 4 to 8 hertz oscillations in the brain. And turns out that these Theta oscillations has a particular role in different cognitive tasks. And this was a review paper in 2019 in Trends in Cognitive Neurosciences. And that shed light that these frontal midline theta oscillations occur when there is a need of increased cognitive control in our brain. So what does this have to do with our short-term memory or memory at all? Here is the graph that I was presenting earlier, and there was the task where people from all around the world, they performed the same task as you did just before, and there was a control condition in which they just had to passively listen to these numbers. And then they measured the brain activity, so the frontal midline theta. And here's the results that you can see. So the blue line, you can see that the memory condition, and the red line, you can see the passive memory condition. As you can see, there was an increasement of the theta power during memory condition, but there was not so much during the listening condition. What's more interesting that this theta oscillation reached a saturation curve, so it saturated after a while, meaning that at a certain point you cannot really increase your theta activity, and that's what people believe that this is the actual limit of the memory load. Here's just another representation of the frontal midline theta. So at the lower panels you can see the memory condition, and as you can see, the more digits you learn or memorize, the higher these theta oscillations are. Now, grab again your paper and pencil, and now your task is to repeat the number sequences you hear, but not in the same order, but in the opposite order. So this is a fail in my presentation. I'm sorry for that. So the instructions are to repeat in the opposite order. Okay. So the first one is 2, 1, 4. The second one is 3, 4, 8, 9. The third one is 9, 2, 4, 5, 6. Next one is 5, 7, 3, 2, 9, 1. Next one is 4, 8, 7, 5, 6, 2, 3. Next one is 1, 4, 3, 7, 8, 5, 9, 2. The next one is 1, 8, 3, 4, 5, 6, 8, 5, 6. The last one is 7, 8, 6, 4, 3, 2, 5, 7, 9, 1. So here are the correct answers. And please go to menti.com and again check your answers and check what was the longest sequence that you achieved to record in the opposite order. Okay, very good. So as you can see, in this task, it requires much more effort, the cognitive effort, than the previous one. So if I were to just compare the two results, the first one was around the average of 9, and here the average was much less. It was 5. So I think it was interesting to see, even if there was a small sample. But let's just dig into what are these differences are between the two tasks. So the one that you have just done, this was called the backward digit span task. This is also part of the neuropsychological tests. So question is that what is the difference between the short-term memory that was the first game about and the working memory that was the second game about? So working memory is kind of the same as short-term memory. Only thing is that you need to manipulate the memory items that you are storing with. So the model, the original model was set up in early 70s, in which researchers they set up 3 different layers in our cognition. One is the phonological loop, one is the episodic buffer, and the other is the visuospatial sketchpad. And the model aimed to and there was also a central executive, so you could see that as a main organization in the brain that kind of allocates different resources to these specific cognitive aspects in our brain. Now, this model has been questioned in the latest literature, and if we were about to just see the taxonomy of attention, it's not clear where this working memory is lies to in the internal attention or to external attention. Now, what do I mean under internal and external attention? So external attention is usually refers to what we allocate to the external environment that surrounds us. For example, the stimulus features like the colors of a tree, or colors of different objects, or the spatial location, for example, where a specific bird is located in the tree. There is also an internally driven attention that, for example, when you were to manipulate these stimuli, for example, if you manipulate the numbers to achieve the opposite order, or to rehearse. So, for example, when you are learning new words, for example, Norwegian words, and then you just repeat within your brain. It's also kind of a internally driven attention. Now, let's see, we developed a new test that can manipulate these two factors, the internally versus the externally driven attention. And the main task is to, people have to remember certain color shape pairings. And there are 4 blocks that has been introduced in this task. And so each of the blocks consists of 100 trials, and the first, or 100 repetitions, and the first 50 repetition contains tasks where the working memory load is kind of low, and the other part of each block contains repetitions in which the working memory load is high. Now, as I said, this test contains 4 blocks, and so each block consists a different number of visual items. So the number of visual items that you see at each repetition varies across the blocks. So in that way, we can manipulate the internally driven attention, so the memory load within each block, and the external driven attention, so or the number of segments also across blocks. Now I want to introduce you another brain wave. Earlier we were talking about theta oscillations. I hope you still remember for that. And now I want to talk to you about the alpha brain activity. So what you can see here in this slide is the normal brain activity that was recorded in a healthy control, let's say. And in the blue you can see where is EEG, so this brain activity when his eyes were open, and in the red you can see when his eyes were closed. What you can observe that when he closed his eyes, there was some rhythmic oscillation going on here, and these rhythmic oscillations are in fact are the so-called alpha oscillations. So alpha oscillation usually occurs when we are trying to filter out the external environment and try to just focus on our internal thoughts. So basically alpha wave functions as Gandalf in the Lord of the Rings when he just filters out the orcs. Yeah, basically it's just you can look at a filter. All right, so what we set up as a hypothesis naturally comes from the nature of alpha and theta waves, so that the alpha waves would refer more like to the external driven attention, and the theta waves would reflect more to the internal driven attention during this specific task that we just designed. I shall give you some preliminary results. So in the next slide, you will see some topographical plots, so over the scope, and you would see the theta oscillations, so how high the theta oscillations were and how high the alpha oscillations were. You would see in the two layers, one direction is the external attention and one is the internal attention. So in the theta oscillations, you could see that the higher the internal attention was, meaning that the higher the working memory load was during the task, this theta oscillation was higher in the, it was more affected by internal attention, but not so much in the external attention layer. However, in the alpha oscillations, that you can see that alpha oscillations were more driven by the external attention and not so much for the internal attention, as you can see. So our hypothesis was kind of seems to be confirmed, but as I said, it's just preliminary results. I would also talk to you some other biomarkers that are not connected to brainwaves, and these are referred to the eye movements data. So people could actually track quite a lot data from your eye movement, and surprisingly a lot. So I'm going to talk about some eye movements data and some pupillometry data. Now, if you are a health person and you studied physiology, I'm pretty sure you remember how the pupil is innerved and how it's organized, its innervation. So basically, if you are not, then the main topic is that you would want to refresh. It's that alpha adrenaline. So adrenaline would just dilate your pupil, so it would dilate your pupil when you are more excited, let's say, but it gets contracted when you are just not that much. All right, so let's see our preliminary results with these tasks. As you can see different lines here. These are how the pupil is changed in the course of one repetition of during the task. And as you can see, this pupil dilation was highly affected by more of the number of sequences, but not so much of the working memory load here. And also, it's not that huge effect here, so it might be just turns out to be nothing. But I still think it's interesting to see. The other thing I want to talk to you about is the fixation disparity that we can also measure if one just looks you in the eye. What fixation disparity is, so let's assume that we have a computer or a screen that we are focusing on. The point or the line when you are projecting it's into the retina, it's called the ISO fixation disparity, so there is no fixation disparity. In case if you were to focus just before the screen of the monitor, then it's called the ISO fixation disparity. And if you were to focus just beyond the layer of the screen, that would cause exo fixation disparity. Of course, this naturally occurs during your life. For example, if you were just thinking something else, then your thoughts wander around and your fixation disparity will get just higher. What you can see here is also the time course of the fixation disparity during one repetition. And as you can see, there's a pretty good effect of the working memory load, so the internal driven attention during the task, but not so much of the external driven attention. We also measure the gaze fixation time at each location of the screen, but it's also not so much, not so important and not so much informative yet. But as I said, the number of samples we have, it's kind of limited. So to conclude my presentation and to give you some take-home messages, the attentional processes are the things that makes the working memory work, and that's what dissociate from the short-term memory. So the difference between the first and the second game was mostly attentional processes. And for the internal attention, it seems that the frontal midline theta is more effective than for the external attention. And also fixation, the disparity, and pupillometry seems to be affected by internal attention. Also, external attention processes tend to be captured by alpha activity of the brain, so alpha oscillations of the brain. And as I said, there is not so. It seems that there is not so much, but maybe gaze fixation time would also capture the external attentional processes. We also managed to develop a new cognitive test that would successfully dissociate these former two attentional processes, and that makes us awesome. I think we are awesome. And by we, I mean this particular department that is located in Motion, called Neuropsychology department. Of course, it was not just a solely work of Helge and Psykose, but we had very good collaborations with the University of Oslo and the several collaborations across Norway. And this project is, or this presentation is more like is in a framework of, as I said, a project called ADHD II, in which we are mainly investigating executive functions and connected brainwaves in adult ADHD II. And part of this project uses this task that I was just presenting here. So this progressive, it's called the progressive acquired equivalence tasks. So if you happen to be a psychiatrist or work with ADHD patients, do not hesitate to contact us. And for now, I would just like to thank all of your attention. And if you have questions, please, now's the time to ask them.
Tusen takk, András. Er det noen spørsmål til András? Jeg ser ikke noen spørsmål i chatten. Jeg lurte på en ting. Jeg regner med at en del av det jeg tenker å gjøre i fremtiden er å koble disse øyebevegelsene mot hjernebølger, og så se korrelasjoner, ikke sant? Har dere noen spesielle planer eller sånne hypoteser dere vil teste ut i forbindelse med det, eller?
Well, it's we are very in the early phase of this setup, but it would certainly be. I do not have a specific hypothesis, but maybe if we were just to see the, and frankly, I'm not so familiar with the results so far, but usually scientists are just measuring the EEG activity and not so much data we have on this specific eye movements data. So it might be just an exploratory analysis in the first place, meaning that they would just set up a model and see the main effects and the effect of the interactions for the eye movements.
Supert. Ok, men jeg ser ikke jeg har fått noen flere spørsmål inn, så da takker jeg bare for en kjempefin forelesning. Så god helg til dere på Nevropsykologisk.
Ja, god helg til alle sammen.
Ja, ha det.
Ha det.